Thanks, Lauren. So I'm really excited to introduce our CCC speaker today, Dr. Dan Larremore. Dr. Larremore is an assistant professor in the Department of Computer Science and the BioFrontiers Institute at the University of Colorado Boulder. He was a postdoctoral fellow at the Harvard T.H. Chan School of Public Health and was an Omidyar Fellow at the Santa Fe Institute. He really has a broad range of expertise, and his research focuses primarily on statistical inference and network science, complex system science to solve problems in biological and social systems. And he's had quite a few really important works across his career. So, for example, a lot of his early focus was on, or I guess I should say, he's got a very continued focus of elucidating just really basic network dynamic properties, particularly one example of that is just efficient detection of communities from network structures. He has a strong focus in understanding social systems of science, so the so-called science of science in a lot of ways. And one of his key papers there was on the inequalities in faculty hiring networks, where he was basically tracking which universities hire faculty from other universities. Another really interesting article, which I hadn't actually seen before yesterday, was looking at how scientific productivity and discovery happen across scientists' careers. So how expected are scientific discoveries? When do discoveries happen across a career? As well as just when scientists are most productive. It's a really interesting article, highly suggest it. He also has a strong background in using these same types of techniques and looking at infectious diseases. So he's studied the evolution of malaria, looking at gene networks and how they contribute to malaria evasion of the immune response. And on a non-science note, I'd say, before yesterday when I was looking through a bunch of his publications, I was still, very much looking at his website, and I highly recommend other people look at his slides on data visualizations, peer review, how to maintain clean code, and also how to give a talk. They are very nicely put together and very informative, and really recommend checking them out if you're interested. So today, of course, he's here to chat about COVID-19. He's conducted quite a few studies on COVID-19 over the past few months, looking at seroprevalence estimation procedures, understanding the idea of immune passports. You may remember when we were talking about, antibody testing and, potentially giving people immune passports to be able to go out and about. And he's looked at how false positives could reduce their effectiveness, looking at mobility correlates with prevalence of disease, vaccine prioritization, and then also what I believe he'll be talking about today is looking at how COVID testing should be working, and constructed a number of important models there. So with that, I'll let him take it away.
Well, thanks so much for the introduction. I really appreciate being here. And I love that briefing in the beginning. I would love to get that as a podcast every day. So I don't know if that's something that you guys are looking into, but I feel like the world would be a lot smarter if we got our information from you guys instead of from, trying to filter out the politics from our Twitter feeds. Anyway, today I'll tell you about some modeling work that I've been doing, and I'll just kind of skip over telling you what the title of the talk is and just jump right in. The picture that we've been thinking about is just kind of this sketch of how viral load progresses over the course of an infection, and how testing, both frequency and the analytical sensitivity, the ability of a test to detect molecules in a sample, could interact with that viral load. So, of course, we have exposure and colonization, and there's some eclipse phase in the beginning after exposure for SARS-CoV-2, where it's basically undetectable at any secondary sites. Concentrations at secondary sites appear to grow, and at some point, they hit the limit of detection of our best tests, the PCR tests from NP swabs or from AN swabs or saliva. And at this point, the virus has really established a foothold, and we get beautiful beautiful mathematically viral kinetics, right? It's basically growing exponentially until it reaches some peak viral load. Symptom onset is usually within a couple days after peak, and then you get a slow decay of viral load until clearance. And so there's some window in the beginning when an individual might be prior to being infectious but positive by PCR. That would be ideally when we would catch them with a test. There's some window during which they're infectious and positive by both tests because viral titers are so high at those secondary sites. And then, of course, some long window at the end prior to complete clearance where somebody still has some of these small microvesicles of viral RNA floating around, and they can be positive by PCR, though they are likely no longer infectious, and that can last for weeks. And so really the idea behind our work is to try and kind of decode this a little bit more mathematically rather than argument by diagram, to try and understand how does the frequency of testing and the analytical limit of detection, the so-called analytical sensitivity, interact in order to tell us something about how we should be testing for public health. And I want to just distinguish ahead of time that medical doctors really care about analytical sensitivity, right? If you go into the doctor and you've had symptoms and you need to know, did I have COVID? Do I still have COVID? It's really important to detect those molecules in whatever sample that you give. But from the point of view of epidemiology and public health, what we really care about is detecting people when they're infectious or prior to when they're infectious so that we can break transmission chains. So in the modeling study that I'll tell you about briefly today, we combined everything that we could find about viral load trajectories. We considered how the assay's limit of detection interacts with those trajectories. And then we considered how the viral load may relate to infectiousness. And then we asked in the model, what would be the impact of some surveillance testing with a particular limit of detection, frequency, and sample to answer turnaround time, the time between you spit in the tube and they tell you whether or not you're positive or negative. So the simple model for viral load is basically just a tent function in log space. So this is log 10 viral load, copies per mil. And these are days since exposure, so exposure on day 0. And we have these 3 control points. They're stochastically chosen. We have a variable delay until the exponential growth regime, a variable peak height and peak timing, and then a variable decay. What we then asked is how would testing interact with this viral load trajectory? Oh, I want to advertise somebody else's preprint. Speaking of living reviews, Mugacevic wrote this fantastic review called SARS-CoV-2, SARS-CoV-1, and MERS-CoV, Viral Load Dynamics, Duration of Viral Shedding and Infectiousness. And it's absolutely fantastic. It took our version 1 preprint model and really elevated it or anchored it much better in all the known data.
So are any of those points data, or are those simulated points? What are we looking at in this?
These are all points drawn from very simple distributions just meant to roughly hit what we know about the duration of the eclipse phase, how high the viral titers get, and how long after the eclipse phase they get. But these are not real data. Yeah. So we asked how testing might interact with these viral load trajectories. So down here is a limit of detection of 10 to the 3 RNA copies per millisample. That's PCR's limit of detection. If you're a PCR person, you know about cycle thresholds. This is a CT count of about 40. Then you have an LOD of about 10 to the 5. This is where an RT lamp test would typically land. So, you know, a 45-minute isothermal turnaround test. And that's about a CT of 33. And so if you imagine an asymptomatic person, don't know that they're infected, on day 6, they get a test, their viral loads should be high enough that we should detect them by either. Then we consider how, go ahead.
Quick question.
Yeah.
So I think you mentioned a term I hadn't heard before. It was something like secondary location. Mm-hmm. And so I assume that means that the virus like infects cells in one location, but then eventually spreads to, you know, wherever we're actually testing it. Can you talk a little bit more about that process, and then also maybe how saliva versus like nasopharyngeal would differ?
So in terms of secondary sites, you know, the places that we look for the virus, these are places that the virus is going to concentrate or at least present itself. So it'll infect cells there. And we need it to be there in stable or sort of repeatable ways from person to person. So regardless of how sick you are or symptoms, what we really want are sites where we can find the virus. And so far, those are basically the NP swabs in the nasopharynx, the anterior nasal swabs up in the nose picking region, and then saliva samples that seem to kind of collect a lot of that and just present it in saliva. I think Anne Wiley from Yale wrote a nice paper that was described something like this: the improved stability and consistency of saliva-based sampling. So with the NP swabs, there's some variability from technique to technique and from sample to sample, whereas the saliva seems more repeatable. But in terms of the biology of like, why does the virus go there and you know, what are the internal dynamics? I actually don't know that much about it.
Okay, so the viral loads you're showing though are like at those secondary sites, like where we would be testing?
Right, right.
Okay.
Yeah, great question. So the next question in the modeling was to ask how diagnosis time relates to infectiousness. And so if you imagine that infectiousness is somehow related to viral load, then if this person got diagnosed on day 6, they would spend days 4 and 5 out in the community, and days 6 until clearance isolated because they now have that positive test. And so in this example, 65% of that infectiousness was isolated and removed. And in the model, we assumed that infectiousness was proportional to the log of viral load. This was consistent with a study from Hay et al. In Nature Medicine. But in the paper supplement, we asked about one extreme, which is what if infectiousness is directly proportional to viral load? So it grows exponentially and then decays exponentially. And then another extreme, which is just that it's binary and it's a threshold kind of on and off. You're either infectious or you're not. So the numbers that I'll tell you about in terms of the frequency required for testing change a little bit, but the gist is basically the same. So we can ask how different testing impacts individuals' infectiousnesses. On the left here, we have no test. And you can see in arbitrary units, kind of the distribution of individual infectiousness. And here's a median as the solid line. And basically, if we test daily, we mitigate all those individual infectiousnesses because we catch people right at the beginning of their viral load trajectories. If we test every 3 days, some individuals are still caught right in the beginning. Other individuals are still out in the community with some amount of infectiousness before they're isolated. And then testing weekly and every 14 days, you can see that these distributions just continue to creep up. One thing that you'll notice is that there's not a huge difference actually between the brown and the pink in terms of the limit of detection of 10 to the 5th, limit of detection of 10 to the 3. So really the main takeaway here is that the frequency of testing and not so much the limit of detection are really driving these differences. But as epidemiologists, what we really care about is kind of how much infectiousness, how much virus is actually out in the population. So we can aggregate over all these individuals, as well as considering what about symptomatic people who may isolate based on symptoms. So this is imagining now infectiousness removed from the population via testing, where we assume that 35% of people are going to self-isolate at symptom onset. So they're behaviorally symptomatic. If we test every day, we basically catch everyone through testing. And very few people are caught through symptom onset because they're caught by testing first. Testing every 3 days, overall, we catch fewer people and remove them from the population at lower rates. And more people, as a fraction, self-isolate because of symptoms. As you can see, this number drops and drops and drops as you test less and less frequently. So if you're testing every 14 days, you're really removing less than 50% of the virus basically from the population. But what we can do is actually ask, well, what does this have to do with dynamics, and plug this into a model. So we plug these viral load trajectories into SIR models, where the E compartment from your typical SEIR model is now subsumed by the fact that you're directly modeling that eclipse phase in the viral load trajectories. And if we assume an R naught of 2.5, then daily testing or testing every 3 days basically crushes the epidemic by pushing R below 1, whereas weekly testing allows a sufficient number of transmission chains to continue that you still get outbreaks of size, around 40% or 50% relative to an unmitigated epidemic with R naught of 2.5. So here's a fully mixed model of 20,000 individuals with constant low importation. We were thinking about CU Boulder here. In the paper, Brian Wilder and Lynn Tambe did an agent-based model in New York City to try and test things with a different model. My office phone rang for the first time in months. Of course it's right now. Okay, so the upshot from what I've told you is that the test frequency really matters for screening and surveillance. But another question that we were curious about was delays. So if I delay the time between the tests and the results, that could have an impact. So same picture as before. What if we have this asymptomatic person who's tested on day 6, but they get the results on day 8? Now they spend 34% of their infectiousness awaiting that diagnosis, and only 31% gets isolated and removed. So down from 65% down to 31%. We can plug those in again to lots of individuals and see how delays impact individuals. So again, across the horizontal here, no test, daily, 3 days, weekly, and 14-day testing. But now these smaller axis labels correspond to the number of days that I've delayed the results in the model. So these 0-day delays are basically immediate turnaround tests. These would be like an antigen test strip or an RT LAMP test. 1-day delay, 2-day delays. And what you can see here is that when I add a delay, it can ruin an otherwise highly effective testing plan at the individual level. So all these blue dots are people who slipped through the cracks and were basically able to complete all of their infectiousness without being detected by a test. And you can see the concentration of blue dots really increases as we both increase the delay between tests and increase the delay between the sample and answer. So there's really no point in screening if there are huge delays. The delays dramatically impact the effectiveness of surveillance. So in terms of infectiousness removed, again, no test, daily, 3 days, weekly, and 14 days with 0, 1, and 2 delays in the turnaround time. And critically, what I want to point out to you in this chart is that for any policy here of how frequently you test, compare a gold standard PCR test with an LOD of 10 to the 3 with an immediate turnaround test at 10 to the 5th. I would take the point of care LOD 5 test over the 1-day delayed PCR test for any of these test frequencies. So in terms of trade-offs or the marginal benefit of speed, we can really sacrifice some of that analytical limit of detection for speed. Just a quick note, here we're assuming 0 contact tracing efforts. So this is surveillance and isolation only. But I hope it's also obvious to you folks who really think about this stuff that if I can get the results back to you today as opposed to 3 days from now, that's really going to be important for contact tracing, both for your memory of your contacts as well as the ability of your contacts to isolate or go get tested. So we can, again, run some simulations. Again, what you'll notice is something like testing every 3 days. If I start increasing the turnaround time to 1 or 2 days, we really see these dynamics crossing that threshold toward an R greater than 1. So the upshot here is that the assay of limit of detection is really secondary to the test frequency and results turnaround time. I won't really go through a lot of these details here, but in the paper we asked about mitigating an ongoing outbreak where we simulate it until you reach some threshold, and then we assume various levels of participation, various levels of testing, as well as sensitivity loss due to poor sample collection. And the upshot here is that the testing can really be thought of as a mitigating tool, not just as something that helps us count the number of cases. In the real world, there are limitations that anybody who's done modeling knows about. Not everyone will want to get tested, particularly if positive tests bring bad consequences. We see this here at CU Boulder, right? Incentives really matter. And if people feel like they're going to get punished when they're positive, you really set up some problems. Positive tests might not cause people to isolate. That was another assumption we made. We know that there are some frat kids here who've been super irresponsible, where they are essentially having a positive COVID test, and then they have a party at their house because they realize isolation means I can't leave, so I'll have the party here, which is just, anyway, irresponsible, I think is the best way to think about it. Misinformation, we know, has had an effect on masks as well as on testing. And there may be economic needs where people just need to go and work because the family doesn't make it otherwise. And so even a positive test might cause them to just go out and work anyway. Finally, if failure to test or isolate or participate in general is homophilic, so birds of a feather flocking together, then impacts are going to be heterogeneous in any of our models. So a fully mixed model won't really be that applicable. Some parts of the community will experience outbreaks when others don't. So just some concluding thoughts here. The punchline is that sensitivity is secondary to frequency and turnaround time. But critically, sensitivity to detecting molecules in a sample is very different from sensitivity to infecting viral load in a person. And so we should really be thinking about the sensitivity of a testing regimen, not just the sensitivity of an individual test. And bigger picture, we do this all the time for medical treatments, right? You don't evaluate an FDA trial based on how well a single dose works in many cases. Instead, you ask, how does the regimen work? And I think we could think about testing the same way. Bigger picture, the within host dynamics matter for modeling. And I think that testing can really help us create a COVID filter where, like the layers of a filter, repeated testing with fast turnaround times allows you to break transmission chains. So with that, just a quick acknowledgement of Roy Parker and Michael Mina, the primary authors on this work. Roy Parker gets a star because he was working this whole time with kids at home. And Sam Zhang in CU's applied math department wrote some really nice online calculators where you can do some sensitivity analyses for yourself. So I realized I was fast and I probably still went over time, but happy to answer questions or discuss.